All right. And in case you're wondering, uh, the sponges are doing the more active filter feeding rather than the passive suspension feeding. Because again, they are feeding their flagella to pump the water through their bodies. So sponges, they of course do not have bones in like the in this in that sense. They don't have a bony skeleton, but they do still have ways of supporting their body and kind of holding themselves upright. There are two main ways. They may have one, they may have the other, they may have both. There are spicules. These are pictures of spicules right here, or rather, they're electron microscope images of spicules. You can see most of them kind of look like little needles, though some look like little spiky balls or even like smoother balls. And these are made of either silica, so basically like glass, or calcium carbonate, something a little more like quartz. And they can be very hard, and they can have kind of these networks of them. And they also make them a little bit less palatable as well, because you don't necessarily want to eat something that's like made of needles. Then they may also, or instead, have spongin. Spongin is a protein. It looks kind of like this. And if you've ever held or squished a natural sponge, then you'll know what spongin feels like. Now, most of these sponges that we have that you buy at like CVS are artificial sponges, but you still can get all natural, like real sponges as well. And of course, before artificial sponges existed, people did use real natural sponges as sponges. So, yeah, spongin and spicules are the two main ways sponges support their bodies. Now, sponges are capable of both asexual and sexual reproduction. And recall, asexual reproduction, you are basically cloning yourself. So you're creating a new individual that is genetically identical to you. And sponges are able to reproduce asexually in a couple cool ways. First, budding. Budding is something actually a few different phyla can do. It's basically where you kind of grow another individual off your body, and that little growth becomes its own sponge. But another really cool one that sponges can do is reproducing via fragmentation. So sponges, you could take a sponge, you can put it through a cheese grater, just grind it up, and each of those little piles of cells will actually like glom together and make a new sponge, which is crazy when you think about it. But again, these cells are like acting pretty independently. So if you just put them in a pile, they'll just be like, okay, and just like glom together and make a new sponge. So very cool thing that sponges can do. They are also capable of sexual reproduction. Most are hermaphrodites, meaning that they produce both sperm and eggs. They are also mostly broadcast spawners, or actually, I think all of them are broadcast spawners. Broadcast spawning is an important term because that's something we'll talk about again and again. Many species are broadcast spawners in the ocean. Broadcast spawning is when you release your gametes into the water. This can be one or both gametes. So basically, for example, corals will throw both their sperm and their eggs into the water. They'll just go, here's sperm, here's eggs, good luck, sperm and eggs. And so hopefully those sperm and eggs meet up in the water and form a new little fertilized cell. Now, with sponges, they actually only release their sperm. So they throw their sperm out into the water, and those sperm get sucked, into another sponge, and then there are egg cells located within the sponge that can get fertilized by that sperm. If you get a fertilized egg and it's released as a parenchyma larva, which is basically a little ball of cells with cilia on the outside, and that will be part of the plankton for a little while before it will settle to the bottom and become sessile and form a new sponge. So this is another example of meroplankton, where it's an animal that is plankton for only part of its life. Now, for many of these groups of animals, I am going to talk about my personal favorite example. And in the case of sponges, those are the glass sponges. So these guys are really cool because a lot of their cells are very transparent. So it kind of allows you to see the structure of those kind of glassy spicules on the inside. So you can see how this like kind of complex network of spicules that make them up. They are typically found in the deep sea, and I just think they're very, very cool looking. Next up, phylum Cnidaria. So these guys, this phylum includes things like anemones, jellyfish, and corals, as well as siphonophores and hydrozoans. They are radially symmetrical, so they have radial symmetry, meaning again, if you like looked at them like a pizza, you could cut them into slices, and each slice would have similar structures in it. They are a little more complex than sponges, but still pretty simple. They only have two tissues in their body, two types of tissue. They have and only two like layers of tissue as well. Uh, and they have no organ system. So they don't have a digestive system. They don't have a true nervous system, anything like that. But they do have some more complex structures than sponges do. And the vast majority of them are carnivores as well. So there are two main body forms when it comes to cnidarians. Where did my mouse go? There it is. I'm trying to drag my little screen over. There we go. So there is a polyp. This is a stage that is attached to the bottom, and its mouth is pointed upwards. And I say mouth, but it's actually a mouth slash anus. And I'll talk about that more in a second. So polyp attached to the bottom, mouth facing upward. And then there's also Medusa stage, which basically you took a polyp and flipped it upside down. So polyp, Medusa. So Medusa is basically, if you think of like a jellyfish, that is a Medusa. So it is a swimming stage and its mouth is pointed downwards, whereas a polyp is something more like an anemone. So these are two main body forms in phylum Cnidaria. Now, like I said, they are pretty darn simple in terms of animals. Their bodies are made of just like three layers of stuff essentially. There's two layers again of tissue. So there is the epidermis, that would be like this kind of dark blue layer here. That is basically its skin, its outermost layer of tissue. Then you have the innermost layer of tissue, which is the gastrodermis. That would be in yellow here, and also in the jellyfish would be in yellow. The gastrodermis is what it uses to digest its food. So its food will go in through its mouth slash anus and then be digested by the gastrodermis. And between those two layers of tissue, they have mesoglia. So mesoglia is basically goo that fills up their body. There are sometimes some cells in the mesoglia, and there can be other stuff dissolved into it, but it's not a true tissue. It really is just like goo. Fills up their bodies. So that is what their bodies are made of: two layers of tissue and some filler goo. They also have what's called a two-way digestive system. So we have a one-way digestive system where food comes in our mouth and comes out the other end. But several of the groups we're going to talk about today have a two-way digestive system where stuff goes in through the mouth, it's digested, and then waste comes back out that same hole. They only have one hole. So that is why this is labeled as a mouth slash anus, because it is both the indoor and the outdoor. It is both. And so it is two-way digestive system. Again, they don't really have organs per se, so they don't have a brain, but they do have some neurons that form a nerve net. So, but they are able to do quite a few kind of cool behaviors, considering that they don't really have a full brain. For example, they're able to do things like vertical migrations, which is pretty cool. This is just another look at the two general body plans from different views. So when you look at them from kind of the top or bottom, you can see how both the Medusa and the polyp have that radial symmetry. Again, if you slice them like a pizza, each slice would have like similar parts in it. This is the lateral view, and this is the kind of sectional view where, again, you can see those three layers that make up their bodies. Now, part of what makes the phylum Cnidaria very unique is their cnidocytes, which are stinging cells. They are located inside a nematocyst, which is the whole kind of structure. And these things are so cool. They're basically little like automated spring-loaded harpoon guns, all covering all the like tentacles of these cnidarians. So this is kind of what an illustration of one. And you can see when they have not fired, they have this little tube coiled up, and they often have a little like kind of barbed tip there, and then they have this hair trigger on the outside. So usually what triggers these is mechanical, meaning like something like brushes up against them or hits them in some way. And as soon as they're triggered, they fire like, again, like a little harpoon gun. So you can see that this spine comes out, the tube gets pulled out, and then through the tube, they can inject venom into whatever it is, whether it's prey or maybe something trying to eat them or just a human that happened to brush up against them accidentally. And these little nematocysts are like automated. So again, the the jellyfish or the cnidarian doesn't decide to fire its nematocyst. It just happens automatically. So that's also why even if you find like a dead jellyfish on the beach, it can actually still sting you because again, all those little stinging cells on its tentacles just fire automatically. So unless it's like started to like decompose, it can potentially still sting you, which is good to know. So these little nematocysts fire extremely fast, sometimes around 700 miles per hour. Again, they really are like little harpoon guns. And they can also be triggered chemically. So, for example, if you often, if we want to watch them fire under a microscope, we'll basically put a little bit of fresh water in with them, and that'll cause them to fire. That's also why if you're ever stung by a jellyfish, you do not want to rinse it with fresh water. You want to rinse it with like salt water or vinegar or something like that because you basically, if you have any little nematocysts on you that have not fired yet, if you rinse it with fresh water, that will make them fire. So you're basically going to get stung more if you rinse it with fresh water. Now, I have a little video here of the nematocysts firing under a microscope. And last time I played this, it caused my Zoom to quit. So I'm hoping it doesn't do that. But if so, I'm just going to start it back up again in another video. So we'll see what happens. But this is, again, under a microscope, and it is very slowed down. So this is like slow motion of the nematocysts firing. So here you've got the surface of something like a tentacle of a cnidarian. They're probably going to add a little bit of fresh water, and then pew, 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 pew. You can see all the little cnidocytes firing. You can see those tubes. You can also see the venom coming out, those little streams of venom coming out of the little tubes. So very cool the way these little cells are able to fire their little harpoons. There goes another pew, 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 pew. Anyway, I think cnidocytes are very cool. Hey, it didn't stop the Zoom. How nice. So Phylum are also broadcast spawners, similar to the jellyfish. When you get sperm and egg coming together, they then form a planula larva right here. That larva then, it's different for different cnidarians. Some cnidarians have 2 stages. Some, I don't know why the video stopped. All right, well, just gonna keep going. It's weird. It's being weird today. Zoom's being weird today. Okay. So you get a planula larva, and there's differences between different cnidarians. Some only have a medusa stage, some only have a polyp stage, although most of them at some point are a planula larva. But some of them have both. Some of them have both a medusa stage and a polyp stage. And for example, many jellyfish reproduce this way. So egg, planula. Then the planula settles to the bottom and becomes a polyp. Then that polyp undergoes what's called strobulation and basically breaks into a bunch of pieces. And each of those pieces becomes another larva called an ephyra, which are actually very cool. They look like little swimming snowflakes. And then that ephyra grows back up into an adult jellyfish. This is another example of meroplankton and what we call a bipartite lifestyle, where they have 2 very different forms that are all kind of part of one organism, which is pretty cool. So, next question for you guys is, we've talked about 2 different species that are both broadcast spawners, and as I said, many species in the ocean are. Would you expect that broadcast spawners to exhibit parental care of offspring? So this could be things like protecting offspring, teaching offspring, things like that. Would you expect that in broadcast spawners? Why or why not? So go ahead and answer that below. Also, I'm going to turn on the light.